Yeah, thank you very much for the introduction, and also thank you very much for inviting me, also the team of Takamasa Miyoshi for organizing this brilliant conference, and also Henry for organizing the session. I'm going to talk about more or less about systems biology, where I'm working in, and I want to tell you something about the frustration with that field, because my interest in data assimilation grew from lots of frustration with models which didn't fit in this area. Some of these frustrations might be known to the people in meteorology or the geophysical sciences. Some of them might be different. So I want to give you just a brief idea of what we're trying to do. And I want to present some method we tried. We also, I'm in the mathematics department, so we also try to develop methods for this field of systems biology, and I would want to present one method we call the dynamic elastic net. But if I would have known more about data assimilation when we started developing that tool, I would have called it probably like a weak constraint variational method, which is very similar to what people did in a different field. It's not identical. I will show there's differences. But so the challenge to give you an idea what the challenge is what I've shown here is some pathways from cancer. And what you see here is a cell, more or less, or a cartoon of a cell. And these little green boxes here, these are proteins. And what these proteins do is they interact via biochemical reactions. And what they do is they transfer information. For example, you have some protein sitting in the cell membrane. We saw already ion channels. There are others. There are receptors. And these proteins get signals by binding ligands from the outside and transfer this information to trigger a response. For example, this could be like a nutrient sensing of nutrients, or it could also be, for example, the growth signal. And this picture here is only about pathways which are relevant or known to be relevant to cancer. This is not the whole picture. This is the signaling pathways which are the most important in cancer. So it means this picture is not complete. And this picture is probably also partly wrong. So because we have lots of uncertainty about who is reacting with whom, we don't really know this network exactly. But this is what you find in the databases. And so what people are trying to do in the systems biology field is try to write down differential equations for these systems by modeling these chemical reactions. And you can imagine this becomes big, so huge. And in order to overcome that, I mean, this problem is old. This becomes huge. People try to model subsystems here. That's what biochemists do for ages by analyzing certain pathways. So they introduce a kind of artificial systems border here and say, well, let me study that guy. I'm interested in these proteins because I know they are involved, for example, in some signaling in cancer or in other fields. And however, I want to argue that this approach is not without problems. And you see that immediately here. If you zoom in here, you see, I mean, if you isolate the system artificially, you will end up with kind of open system, because this pathway here, which is a MAP kinase signaling pathway, it's a pathway of phosphorylation steps here, very well studied in a way, talks to other pathways. And you can never be sure that these influences can be neglected. So you have essentially an open system here. In addition, there might be also feedback loops. So for example, the output of this pathway might feed back somewhere into this system. And this kind of illustrates the challenge here. I mean, model building is a hard, difficult process. People know that from meteorology. However, we have some advantages in biology because we can do experiments. So we can do lab experiments, which is hard in the weather field, of course. However, from the many experiments the biologists can do, it's not easy to find out which one is the right one and which one is the most informative one. And one task for a computational biologist like me is to tell them what is the most interesting experiment. So, and in order to try maybe to build models of these things, we had some experience with several different fields, and we came up with a kind of thing, yeah, we need something to deal with this uncertainty here. So the uncertainty, as I told you, is we don't really know these reaction networks in detail. We don't know all the details. We know a lot, but not everything. We don't also know the details of these chemical reaction rate laws, what we call, I mean, the dynamic laws of these reactions. We have some assumptions we can make here. However, we can never be sure whether those rate laws are true. We don't know the parameter values, and parameter estimation is a big field in the systems biology area. However, nobody in the systems biology area knows the term data assimilation, or nearly nobody. Yeah? That's a funny thing. So, and I think my personal opinion is that this data assimilation will play a bigger role in the field. And what we usually have, we have concentration measurements. So we measure the concentrations of some of these guys, of these proteins over time. For example, protein concentrations can also be RNA concentrations. And however, not all of them can be measured. So we have incomplete measurements. This is the same in metrology. We have an observation equation or measurement equation. However, some of the proteins, even when they are there, can often not be seen. Because, for example, mass spectroscopy experiments, sometimes some proteins, you know they are there, you can't identify them. So there's lots of noise and uncertainty in here. So what we need is kind of methods for modeling under these like very uncertain and incomplete information conditions. And we came up with an idea how we can kind of like estimate state, do state estimation, even in systems which are not perfectly, or which are not perfect models. And just to introduce some notation here, so what I consider is a just standard state space model. You have some state vector, which are, in our case, usually concentrations or abundances of proteins consisting of n protein concentrations. You have measurements, usually m measurements, which are usually m is much smaller than n. And we like to talk about networks. We saw that before in the neural field. So this network here represents, in a way, this differential equation. So because we like to draw so-called influence graphs, and the idea is that you have for each state variable, or each protein in our case, you have a node or vertex, and you draw an arrow from one vertex to the other if the rate of change of that vertex is influenced by that one, and formally means that the jth component of f, the partial derivative, is non-zero. Yeah. So these influence graphs. Of course, you have also inputs. These green ones are known inputs, and we have the measurement function is h in my notation. Sorry for maybe my notation is different from what is used in the metrology field. And what you also do is, of course, you have the system sports here. And what you usually do when you develop a model is you have some draft model, what we call the nominal model system, which takes into account a certain set of interactions. So you put everything you know or you think is relevant for your pathway into this model. However, when your data don't fit or don't make good predictions, or when your model doesn't fit and the data doesn't make good predictions, then you know that you have missed something. So that means the true system has probably more state variables which kind of interact with the state variables in your system. And you might also have interactions between the state variables within your system which you haven't covered because you don't know about these reactions, for example. So, and the typical approach in systems biology is, yeah, well, then take these guys into account, makes the model bigger. And this leads to big models, lots of parameters, not easy to handle. And what we think is probably a smarter way of doing that is to keep the simple model, which covers, because this is maybe the pathway or the bit of the network you're interested in, and treat these model errors here as hidden inputs. That means the red arrows here, these red curly string errors, are hidden inputs, and we represent them. We have hidden inputs everywhere where we have interactions which we haven't covered in the nominal model in comparison to the unknown true system we postulate. So the red ones are the hidden interactions. And so the model error is very similarly defined here, just for the notation. So we say, look, this is our model draft. We have our systems model. I put a tilde on top because this model is still a bit shaky. We know it's not really true. But we assume here that we know the output function relatively well, which is a reasonable assumption in systems biology. However, the true model would have another right-hand side, another vector field here, but we can represent it as the vector field of the nominal model plus a model error, W. What we saw that before in this conference here. So for the definition of a model error is just that it's the difference between the rate of change of the true state space trajectory minus the vector, the rate of change of the nominal model along the true trajectory. That's my definition of model error here, to be explicit. And the goal is now to use data to estimate that model error. And so what we have is this nominal model, the draft, and we have data. As usual, we have like measurements for some proteins or for combinations of protein concentrations at certain discrete time points. And what we want to know is, we want to know where is our model wrong. That means we want to know which components of this model error vector are non-zero, which we have in this set here, W. Also interested really in the model error itself, because this model error might give us ideas about interesting biology we don't know yet. So we want to infer the dynamics of this thing. And we want, of course, have a state trajectory, or we want to know the state of the system. Even when the model is wrong, we want to know the state of the true system, not of our model. Yeah? And of course, this is an ill-posed problem. It's an inverse problem, because what could happen is that different models or different hidden inputs or model errors could generate the same data. So what we call that, because I've also some ideas, background in control theory, the non-observability of the model error. And I will show you later that this is a serious problem. To alleviate this problem a tiny bit, we make additional assumptions. So the one assumption is the usual assumption for this model error signal. We assume that this is a smooth function. This is the same as in weakly constrained 4D war, as people would call it here. In addition, and this is different, we assume that our model error vector is a sparse vector. That means it contains many zeros. And this assumption is here that you can justify that assumption from different perspectives. One is we only want to see the gross errors, the really big ones. We're not interested in the noise here. Or in addition, we want to also assume that our model is not everywhere totally wrong. Yeah? That's the idea here. And we put that into what I call an optimal control problem. People here would call it a variational problem, I think. So we say, look, we have these measurements, y, at certain time points. And we take our nominal model here and add this unknown hidden input or model error here. I put hats here because this is used for estimation. We call that an observer system. This is a term from control theory. And we have, as usual, we have an objective function, which we minimize with respect to the model error here. And this has two contributions, this function. The first one measures how well the model fits, how well the output of the model or the observer system is aligned with the data. So this is quadratic term. You can have here like scale that with the variance, covariance. This is usually not a big problem in systems biology, this covariance here. However, so this measures how well the data fit, and we have a regularization term. So, and this regularization term has two different terms here. One term is a L2 norm, totally what people do here in the field. That means, and this makes the signal smooth, yeah? So it you prefer solutions which are smooth. But in addition, we have here an L1 regularization. That means we put here a second term in. And if you have a little bit background in high-dimensional statistics, you know this is kind of what the lasso people did in regression. That means you have here the L1 regularization makes this signal sparse. So it kind of encodes the second assumption that our model error has many zeros. And we can weight these different contributions by these two parameters, alpha 1 and alpha 2, which are regularization parameters. And we can, it's not that difficult to adjust them. So I could talk about that, but I will, we found it's easy to find these parameters which are reasonable. It's not very sensitive to that. So then we minimize that and estimate the model error at the same time estimate the state. So in a way, very similar variational method. And to show you some examples how this method can be applied, I start with a very simple model from Swarmai, already a bit older, which is about the EPO receptor. Some people might know that from Lance Armstrong. That's one guy he wanted to have more from to be faster in cycling. So what it does is the EPO molecule or EPO ligand binds to this receptor and triggers a signaling cascade, which consists of 4 proteins, which are called the step proteins, but I call them here X1, X2, X3, X4. And then this signal triggers the expression of a gene or the activation of a gene, cellular response. That's not that important. And these people, the Swarmai people measured data, but they found out that this model they came up had one structural error. After quite some tinkering, they found that they missed the feedback loop here that the last protein, which is in the nucleus, is recycled here. And this was important to fit the data, otherwise you can't fit the data. We asked, could our method, could the dynamic elastic net have learned that from the data? That's the question. So could we just take the model without feedback as a nominal model and learn that from the data? And here's what we did. So you see here the nominal model. That's just the cascade of reactions with a known input, which is this signal here, which is a stimulation of the receptor. And then we have data here. We have 2 outputs, which are combinations of these proteins. Because, for example, you can't distinguish phosphorylated and unphosphorylated forms here. These are these data points here, the black lines. And you see that the nominal model, these are the blue lines, cannot fit these data, even with hard parameter fitting here. So that means there's structurally something wrong. So then we apply our dynamic elastic net. So we assume these hidden inputs here are model errors and apply it. And then we get this signal here. We estimate the W1 hat and what W4 hat, what W3, and so on. Here you see also the area under these curves to see which are the biggest signal. And you see that W1 and W4 are the dominant signals. There's a tiny bit signal of W3. However, this is consistent with their model saying that there is a feedback from 4 to 1. So we could recover the target nodes. That's our goal. We want to know where the model is wrong. And it seems to, at least when we assume that this is the ground truth, that to work here. In addition, we can also modify the state estimates, as you can see here. Here the blue lines are the state estimates from the nominal model without feedback. And here you see the modified state estimates for the concentrations of these 4 proteins, which we couldn't measure directly. So we have a data assimilation with model error. Fine here. So, but this is a simple, relatively simple model, very small. So we tried that for a bigger model from Ouyang, 2014, which is a model for how plants respond to ultraviolet light. So it doesn't really matter what it's good for. It's bigger, it has 11 protein concentrations, 10 reactions. And we induced artificial model error because we wanted to know the ground truth here and ask whether we can recover this error signal and also the true state. That's our goal here. So here you see the inference graph of the model. Here, in this case, we applied the model error to the nodes X3 and X9. And we have 5 measurements here, which are either directly concentrations or combinations of these concentrations. And you can see the nominal model, the blue lines, cannot really fit these data sufficiently because of structural error or systematic error here. By applying the dynamic elastic net, putting hidden inputs to all these nodes here, we can fit the model, the data, which is not really sufficient, and we can also recover, this is W3 and W9. We can also, here you see the error in the estimates. It's pretty small in the estimates of the model error, so we can also roughly recover the dynamics. And we can also recover the state, because the state error is around 0 here in comparison to the other model. So, and then we tried to do all the pairwise combinations of this. And this table you see, the rows and lines are combinations of nodes. For example, when we perturbed node 9, we recovered node 9. When we perturbed node 8 and node 9, we recovered node 8, 9, and node 8. However, there were some mistakes made. And for example, by applying the model error to node 1 and node 9, we recovered node 9 and 3. You see that here, 3 and 9 are the biggest signals. And we call that a non-observable model error, because this is a similar explanation. So we can't recover the truth here, because there's another possibility which generates the same outputs. And then we said, OK, when we can't recover the one single solution, we try to find more and give the biologists like a ranking of different solutions. Then they can test that experimentally. That's the idea. And we came up with a simple algorithm to explore suboptimal solutions. The basic idea is to, once we have found this solution, and we don't know whether it's really true, we constrain, for example, W3 to 0 then, and then run the algorithm again and look whether other sparse solutions pop up here. And then, in this case, this was the second highly ranking in terms of the objective function. And we can go on and then get kind of like a ranking of solutions, and then biologists can go and find it. So I will skip the next section, because this is essentially this algorithm a bit more formally. So in order to summarize, I say, well, I present you the dynamic elastic net. Probably should have called it L1 regularized 4D, weakly constrained 4D var or something. So we didn't actually know about this algorithm before. And what it does, it tries to estimate the nodes in the network where the model could be wrong, or gives you at least ideas, and does state estimation for incorrect model. However, this only works when your model error is observable. And I think this is a key issue here. If it's not observable, meaning that if there's no unique solution, then I gave you an idea where we have an algorithm which can explore other solutions, which can then give hints to the experimentalists to test experimentally. What we are currently doing is we also try to better understand which model errors can actually be observed. And can you see that just from the network, from the structure of the network? And there's some theory about that in control theory. And we are working with that. That means we can just figure out all the configurations much faster than we do here and go to bigger networks. That's our goal here. We also want to use that to design experiments to improve the observability and also to find out what are the right models here. And we also want to assign uncertainty or confidence intervals. We already have a method. We made this method, namely, we made it a Bayesian method now. In terms of the path integral formalism, this could be the minimum or the Laplacian approximation. And then you can just make it a probabilistic thing. And to give you an idea, for example, this again for the stat thing, you could get, for example, like intervals, confidence intervals, or for these state estimates and so on. So, but I didn't talk about that, but we have something submitted for that. Yeah. I'm at the end. I want to Holger Fröhlich and Benjamin Engelhardt, both from the University of Bonn, and also Holger is a principal scientist at UCB Biosciences in Germany. And people who want to read more, they can read our paper in Scientific Reports. Thank you very much.